Welcome back. In this lecture, we look at the microbial phosphorus cycle as part of the elemental cycles module in this MOOC. Unlike carbon, nitrogen or sulfur, phosphorus has only one stable oxidation state. That means it's not involved in redox reactions and does not contribute directly to microbial energy generation. Phosphorus is essentially only present in the form of phosphate. Microorganisms use phosphate almost only in anabolic processes. To build cellular structures and biomolecules in nature, phosphorus doesn't undergo redox conversions in a way as carbon, nitrogen or sulfur does. It mainly changes its form from being soluble, adsorbed to minerals, precipitate or bound in biological material. Phosphorus is often the limiting element for life in ecosystems. For human society, phosphorus is one of the most critical non-renewable elements. We depend on it for food production and it cannot be replaced by any other element. It's good to realize that there are rare cases of phosphate reduction to phosphine gas. However, this process is extremely limited in nature. Now, let's go through the main processes in the phosphorus cycle. First, we have immobilization, the uptake of phosphate by microorganisms of plants to produce new biomass. Second, there's mineralization. When bacteria decompose dead biomass, they release phosphate and other nutrients back into the environment. Third, we have solubilization. Phosphate in soils is often bound to iron, calcium or other minerals, making it unavailable. Certain bacteria can produce organic acids or chelating compounds that solubilize mineralized phosphate, making it accessible again to themselves and to plants. Next, phosphate can precipitate or absorb onto mineral surfaces. While this is largely a chemical process, microbial activity can influence it. Finally, phosphate can be released from sediments back into the water column, for example in lakes where phosphate is bound to oxidized iron. If microorganisms reduce the iron from its oxidized to its reduced form, the bound phosphate is released and becomes available again in the water. Phosphate plays a central role in biology. It's a crucial component of several key molecular groups. First, ATP or adenosine triphosphate, which is the universal energy currency of the cell. The phosphate ester bond in ATP is a high energy bond that drives most cellular work. Then there are DNA and RNA, the carriers of genetic information. They have phosphate groups in their backbones. Thirdly, the phospholipids, which form the cell membrane. Without these membranes, the cell could not maintain a separate internal environment, distinguishing it from the external environment. Then NADH and NADPH, which act as the main electron carriers in the metabolism of cells. They also contain phosphate and are vital for redox reactions and energy transfer. And finally, various phosphorylated intermediates act as metabolic regulators and intermediates in biochemical pathways. So, while phosphate doesn't provide energy directly, it's fundamental to how energy and information are managed inside the cell. In environmental biotechnology, phosphate removal and recovery are very important. When phosphorus is released into the environment, it can cause eutrophication, the excessive growth of algae and microorganisms in water bodies. As this biomass decomposes, oxygen is consumed, leading to oxygen depletion and the death of aquatic life. If nitrogen is limiting, nitrogen-fixing organisms can still grow. But if phosphate is lacking, no growth is possible at all. That's how essential it is. Phosphorus is also a non-renewable resource. We currently mine phosphate rock, but global reserves may be depleted within the next 50 years. That means recovery and recycling of phosphorus are crucial for sustainable food production. In wastewater treatment, phosphate removal can be achieved by chemical precipitation or biological accumulation. Certain microorganisms can store phosphate inside their cells as polyphosphate, allowing for both removal from the wastewater and potential recovery of the phosphate. So what's the role of polyphosphate storage for microorganisms? Almost all bacteria can store phosphate as polyphosphate granules. They do this to buffer against phosphate scarcity in their environment, ensuring that they always have a reserve for growth when external phosphate is limited. A smaller group of bacteria can also use polyphosphate as an energy reserve. These are known as polyphosphate accumulating bacteria. They thrive in dynamic environments, where conditions alternate between anaerobic and aerobic. For example, in estuaries that flood and drain with the tides. At high tide, they are anaerobic. At low tide, they are aerobic. Under anaerobic and nutrient-rich conditions, these bacteria take up organic substrates. To do this, they use energy stored in polyphosphate, breaking it down, resulting in the release of phosphate in the environment. When conditions turn aerobic, they use the stored organic material for growth and rebuild their polyphosphate reserves, preparing for the next anaerobic phase. We apply this principle in wastewater treatment by creating controlled environments that mimic these alternating conditions. In the anaerobic tank, wastewater containing organic carbon is mixed with the microbial community. Here, polyphosphate accumulating bacteria take up volatile fatty acids and store them as polyhydroxyalkanoates or PHA inside their cells. To convert volatile fatty acids into PHA, they need both energy and reducing power. The energy comes from breaking down polyphosphate, while reducing power comes from the degradation of glycogen to PHA. In the subsequent aerobic tank, oxygen or nitrate is supplied. The bacteria use the stored PHA for growth and energy, and at the same time replenish their polyphosphate and glycogen reserves. Finally, the biomass settles in a clarifier and the cycle begins again. Under an electron microscope, these organisms show dark, dense polyphosphate granules with clear PHA inclusions and lighter regions corresponding to glycogen. Modern wastewater treatment plants are designed to remove carbon, nitrogen and phosphorus simultaneously. Although the detailed configurations vary, the key principles remain the same. We enrich microorganisms that can perform biological phosphate accumulation so that phosphorus is removed from the wastewater. At the same time, other microbial groups nitrify ammonia to nitrate and denitrify nitrate to nitrogen gas, completing the nitrogen cycle. Because polyphosphate breakdown releases phosphate under anaerobic conditions, and take up this released phosphate plus the phosphate of the influent during the aerobic phase. The liquid in the anaerobic tank therefore contains high phosphate concentrations. This phosphate-rich liquid can be treated separately, for example through chemical precipitation to recover phosphorus as usual product, before returning the liquid to the main treatment system. This way, we not only remove phosphorus, but can also recover and recycle it. To summarize, phosphate itself plays no role in microbial energy generation. There is no oxidation or reduction of phosphate compounds. But it's vital for the cell's anabolic metabolism and for the energy transfer system through molecules like ATP. Polyphosphate storage provides microorganisms with both a phosphate reserve and, in some cases, an energy buffer. In environmental biotechnology, polyphosphate accumulating bacteria are key for phosphorus removal and recovery from wastewater, an essential step in sustainable nutrient management and protection of our aquatic environments.